You're right. Thank you for letting me know. All right, so today we're covering chapter 4. Let me share my screen. Know the terms. We're going to be talking about how gram-positive and gram-negative cell walls differ, and then describe the importance of taxis in bacteria. That didn't move. There we go. So when we're looking at prokaryotes and eukaryotes, you should know that prokaryote actually is Greek for pre-nucleus. Literally, that would be pre-kernel, and the kernel is translated as nucleus. Eukaryotes, it's Greek for true nucleus, true. Prokaryotes have one circular chromosome. It's not in the nucleus. Eukaryotes generally have multiple chromosomes and they're linear, meaning they have a starting position and an ending position, and the chromosomes are in a nucleus. The DNA of prokaryotes is not associated with histones. Histones is a protein, and the DNA of eukaryotes is wrapped around this histone protein. And so DNA of eukaryotes and also of prokaryotes does have, let's see, not the DNA, but the chromosome is made up of DNA and protein. In eukaryotes, the main protein is histone and the DNA wraps around it. Any question about any of that? Prokaryotes have no organelles, and by organelles, I mean membrane-bound organelles. Prokaryotes do, and that would be the mitochondria, the chloroplast, the Golgi apparatus, and we'll talk a little bit more about organelles later in this chapter. Prokaryotes have peptidoglycan. Actually, I should say bacteria have peptidoglycan in the cell wall, and it's the main molecule of the cell wall. Archaea generally do not have peptidoglycan. In eukaryotes, none of their cells have peptidoglycan, even the cells that have cell walls. Prokaryotes, their cells divide by binary fission. Usually a cell simply splits in two. That's binary fission, to divide from one cell into two. Eukaryotes divide by mitosis. Their cells divide by mitosis. Any question about any of that? All right, so we're going to be talking about prokaryotes, discussing the cell shape and arrangements. We'll talk about cell structures of prokaryotes, and then the movement of material across membranes, which in prokaryotes, the only membrane is the cell membrane. Then we'll talk about eukaryotes, which you should have had before. Excuse me. So if I don't finish that, I will just skip it because you should have had this in your preliminary course. But we'll try and talk about the cell structures of eukaryotes. And then we'll end with the endosymbiotic theory, which is a theory for how eukaryotes came about. All right, when we're talking about prokaryotes, they come in different sizes and shapes. The average size of a prokaryote really is about 1 to 3 micrometers. 10 micrometers is quite long for your prokaryote, but the average is about 1 to 3 micrometers, and that's the diameter. Some of the long ones can get up to 10 micrometers. The basic cell shapes are cocci, which is plural, coccus is singular, and they're spherical or round cells. And then there's a bacillus, which is singular, bacilli, which is plural. And we also call these rods. rods and bacilli, those two terms are used interchangeably. However, the round ones, we never call them round ones. We call them cocci, or coccus if it's singular. And then we have the spirals, which are sort of S-like shape. These are the three basic shapes of all prokaryotic cells. If a cell has another shape, it is generally a shape based on these three basic shapes. There are some exceptions, and we'll talk about some of them, but most bacteria come in these three shapes or a derivative of these shapes. Like you can have a Coccobacillus, which is in between a cocci and a bacilli. Any question about any of that? When we're talking about spirals, there are different types of spirals. There's the spirillum, which is the regular spiral. And then there's a longer, more, I don't know, wavy or S-shaped spirochete. These two actually differ in that this one is more flexible and this one is more rigid. And then I don't know if you want to call this a spiral or a curved rod. A vibrio, you could say it's half of a spiral or it's a curved rod. So a vibrio is a curved rod. We're not really going to talk about vibrios, and I generally call them a curved rod. There are some unusual shapes, like Stella is a star-shaped bacteria. Haloarcula and Archaea is, it's kind of squarish or rectangular. That's clearly a rectangle. But most of them are cocci, bacilli, or spirals. Most bacteria species are monomorphic, meaning all of the cells have a single shape. Like all of these are rectangular. But a few species are pleomorphic, where the cells take on many different shapes. Like this species here is pleomorphic. Any question about that? When we're talking about prokaryotic cells, you can talk about the cellular arrangement, sometimes called the cellular growth arrangement, or maybe just the growth arrangement. And that is how the cells stick together. When we're talking about cellular arrangements, please understand that you might have singlets, a single cocci. You can have diplococci, and that's two cocci stuck together. And then you can have streptococci, and that would be cocci in a line. Cocci can also take tetrad shapes, 4 of them stuck together, or Sarxinia, that's 8 of them stuck together. And then they can have staphylococci, which is a cluster. And this looked to somebody like a cluster of grapes. So staphylococci actually means cocci in a shape like grapes. That's what staphylococci mean. Any question about that? The cellular growth arrangement.
I have a question.
Okay.
It's about the previous side. When you were talking about pleomorphic cells, do you mean like there could be one species and one cell might be like this beryllium, but another cell of that same species might be like the rod shaped?
Yes.
Okay.
Usually when they're pleomorphic, they look more like this, meaning they do take on different shapes. But there is one species, I don't remember the name of the species. I used to know that, but there is one species which actually is pleomorphic in that it has spirals, or the cells can be rods. Where do I have that? There they are. So that would be pleomorphic in that there's spirals and there are rods. There's actually very few species that have that combination. We're not going to talk further about that species. I remember if you put that species in liquid culture, most of them are spirals. But if you grow it on a solid media like agar, most of them are spirals.
Okay, thank you.
Okay, but most of them that are pleomorphic look like this, and they have many different shapes. And the reason why this one is pleomorphic is because it doesn't have a cell wall. And so the shapes of the cells vary because the cell membrane takes on a different shape. You don't need to know that, but just telling you. All right. The cellular growth arrangement comes about because of the plane of division of the cell. Like diplococci and streptococci, the cocci always divide on this plane. And so they always go this way. And the diplococci breaks after 2, and the streptococci doesn't break. And so a new cell just gets put on to the growing chain. And then the tetrads have 2 planes where the cells divide. They can divide on this plane, or they can divide on that plane. And that gives rise to a tetrad. And the Sarxinia, as well as the staphylococci, the cells can grow on all 3 planes. So they can, well, divide on all 3 planes, meaning X, Y, and Z. Any question about that? Now, bacilli have some of the same arrangements as cocci. You can, of course, have a singlet bacillus. There's one there. You can have diplobacilli, and that's 2 hooked together in a hot dog-like connection. If the link is on the long side, we don't call that diplobacilli or streptobacilli. We call that a palisade. And if you turn this palisade on the end, so one end is on the ground and one end is up, it looks like a palisade fence, which isn't very common anymore, but you just put a board, stick it in the ground, and then attach another board to the side. And that used to be very common. And if you remember reading Mark Twain, I think it was Tom Sawyer was painting a palisade fence. Anyone remember that? Okay, so those are the only growth arrangements that bacilli have. Singlets, diplobacilli, streptobacilli, and then palisades. All of the other cellular growth arrangements, bacilli do not form. And then here is Ococcobacillus. It is a cell in between a coccus and a bacillus. And there we're looking at them there. Let me blow that up. So that one there is just an elongated coccus. And that one there, let's go to that one there. That one is a real short rod. Okay. And that's what a Ococcobacillus is, in between a cocci and a bacilli. Any question about that? Let's go back one slide. The spirals only take on a singlet, meaning with spirals, the only growth arrangement is a single spiral. They have no other cellular growth arrangements. All right, so this slide is showing you some of the structures of a typical prokaryotic cell. All of the structures shown in red are structures that all cells, well, I should say all prokaryotic cells have. All cells have a cytoplasm, ribosomes, and a plasma membrane, as well as DNA. Prokaryotes have a nucleoid region. Hopefully you're not seeing that. The nucleoid region is where the DNA is in the cytoplasm of the cell. And the DNA is just in solution in the cytoplasm. And wherever the DNA is floating, we call that the nucleoid region. There is no boundary between the nucleoid region and the cytoplasm in a prokaryotic cell. And the DNA can just float and move around in the cytoplasm. But it looks different when we stain the cells. Even in the electron microscope, it looks different. And so that would be the nucleoid region. And it's just the DNA takes on a little different staining properties or characteristics than the rest of the cell. Any questions about the nucleoid region? And the nucleoid region could be anywhere in the cytoplasm. It's just where the DNA floats. The other structures are seen in some prokaryotic cells, like a flagella. Not all cells have them. The fimbriae, not all cells have them. I'm not sure why that isn't red. Oh, because it's right up here. Cell wall. Actually, not all prokaryotic cells have the cell wall. Most of them do, but not all of them do. The capsule, not all of them have. We'll talk about inclusions in a little bit. The glycocalyx or glycocalyx, both pronunciations are correct, is something that is secreted by some cells. Many prokaryotes, but not all prokaryotes, secrete a substance outside the cell wall. It's usually sticky, and the main molecule of the glycocalyx is the water. There are three types of glycocalyxes. If you can see it with the light microscope, it is called a capsule. A capsule is neatly organized. It has more molecules than just water, meaning there are other molecules in the capsule. And the capsule is important to the cell because it can prevent phagocytosis. And so generally a capsule will make a cell more, what's the word I'm trying to say? More pathogenic, because the capsule prevents the white blood cell from phagocytizing the bacteria. If you cannot see the glycocalyx under the light microscope, it is probably a slime layer. The slime layer is unorganized and it's a loose attached structure, and the main molecule is water. And that's why you can't see it with the light microscope, because all it is mostly just water. You can see the slime layer if you use the electron microscope. And where did we see that? I thought I had a picture of that. Right there is a little bit of a slime layer. Oops, went too far. The sticky stuff, let's see if I can blow that up a little. The sticky stuff right there is the slime layer. So if you can see it with a light microscope, it's a capsule. If you can't see it with a light microscope, it's called the slime layer. Both of them are glycocalyxes. And then there's a third type of a glycocalyx, and it's only found in biofilms. We'll talk about biofilms later in the term. It's also called, this glycocalyx is called an extracellular polysaccharide, or EPS. And it's something the cells secrete to make a structure around the cells. Any question about any of that? All right, if a bacteria can swim, then it has a flagella, because that is the only structure that bacteria and archaea have for locomotion. Now, eukaryotic cells can move by flagella or by cilia, we'll talk about that, or by, what is those called? Not phagosomes. Phagocytes? I'm drawing a blank on what that's called. What blood cells use to move, and that is they put out the cytoplasm in one location. We'll talk about that later. But bacteria, if they swim or move, they only do so with a flagellum. The flagellum is largely outside the cell wall, and it's a whip-like structure that actually moves, it beats, and that's what gives the cell the motility, the ability to swim. But you should realize that there is a little bit of the flagellum which goes through the cell wall, so it is attached to the cell wall, and in fact, it's also attached to the plasma membrane. Flagella are made of protein, and the main protein is flagellin. Any question about any of that? You can have 4 basic arrangements of how the bacteria are arranged. On one side, on both sides, multiples on both sides, and then all over the place. I'm not going to quiz you on the names, so you don't need to know the names, but there was a lab question that asked you about this. So if a bacteria cell rotates or swims, it has a flagella, giving it the ability to run or tumble. By tumble, we're meaning it sort of, I don't know, tumbles like a barrel in the solution. Taxis is the movement of cells toward or away from a stimuli. This right here is actually movement away from the light source. So these cells are not photosynthetic, they're moving away from the light. And that is taxis, movement away from a stimuli. Movement towards the light would also be taxis, and both of these are phototaxis, movement towards or away from light. There's also chemotaxis, and that would be movement towards or away from a chemical. Any question about any of that? Now, I mentioned that bacteria, if they move because of a flagellum. However, the spirochetes have an unusual flagella, and we call it an axial filament or an endoflagella. I use the term endoflagella. It's only seen in spirochetes, where the flagellum is anchored at one end of the cell, and it is inside the cell wall. So let me blow this up. Scroll down. That's not very good. Hopefully you can see it better now. So here's the cell wall, the light pink. And you can see that the axial filaments, or the endoflagellum, is inside the cell wall. And that axial filament or endoflagella actually wraps around the spirochete. And there we see it in an actual electron microscopic image, a scanning electron microscope, where the endoflagella is wrapped around the cell. And it's very hard to see, but that light pink is the cell wall. And so the endoflagella is inside the cell wall. Any question about any of that? Anyways, the spirochetes use this endoflagella. Oops, that's not what I wanted to do. I wanted to open it. There we go. The spirochete uses this to move, and it gives a very unusual corkscrew-like motion. Oh, I don't know what's wrong here. That's not the link. Come on. It's something with this browser. It's not allowing me to paste. Oh, there it goes. Five seconds, we have to watch the commercial.
2022 is your last chance to go solar. In December of 2020.
So there's the spiral-like movement. And you notice how it can just change its direction. And then let's go back just a little bit. You see how the cell changed from going this way and that way to moving that way. And the spirochetes are very flexible. Any question about that? And just by rotating one way, it'll move that way. And then to go the other way, it just rotates the other way, meaning moving in a corkscrew-like motion. Any question about any of that? Okay, fimbriae are hair-like structures attached to the cell. They are thinner, shorter, and straighter than flagella. The flagella is a structure used in locomotion. The fimbriae is a structure that's used for attachment to surfaces. It is not used for swimming. Any question about any of that? Fimbriae can be pathogenic, and that is the Fimbriae may allow the pathogen to attach to our cells.
These structures that we're talking about are only in prokaryotic cells, correct?
Correct.
Okay.
Although the glycocalyx and the cell membrane can be in other cells, meaning in eukaryotes.
Okay,
But
thank
most of
you.
them are only found in prokaryotes. All right. The pili, also called the sex pili, is a structure similar to the Fimbriae, but it's longer. See there, actually you can see the Fimbriae right here. Blow that up. This is Fimbriae right here. And the sex pilus is a little longer and thicker. It's also hollow. So it's hollow inside. We'll talk about the sex pili later in the term. It's a structure used to transfer DNA from this cell into that cell. And for your purposes now, you just say the sex pili is a structure for transferring DNA from one cell to another cell. Any question on any of that? All right, let's talk about the prokaryotic cell wall. It is a complex, semi-rigid structure, and the cell wall is responsible for giving the cell its shape. The reason why a cell is the shape of a coccus or the shape of a rod or most other shapes, unless it's pleomorphic, it has its shape because of the shape of the cell wall. The cell wall is complex. It's a semi-rigid structure. Its primary purpose is to protect the cell and prevent the cell membrane from blowing up and destroying itself. Meaning, if the cell did not have a cell membrane, water could come into the cell and blow up the cell membrane. More water coming into the cell would blow it up even further until eventually the cell membrane would burst. And then the cytoplasm would leak out and the cell would die. Excuse me. The cell wall prevents osmotic lysis, so that with the cell wall being there, water can come into the cell, and then the cell membrane can expand, but only up to the cell wall. Once the cell membrane reaches the cell wall, it cannot expand any further because the cell wall doesn't move, it's rigid. And then that protects the cell membrane by preventing any further water from moving into the cell. And then the cell membrane cannot expand and it won't burst itself. Any question about that? The cell wall can contribute to the pathogenicity in some species, like mycobacteria. The cell wall does contribute to the pathogenicity. The cell wall is mostly made up of peptidoglycan, at least in bacteria. Peptidoglycan is the main molecule in the cell wall of bacteria. Let's talk about peptidoglycan a little more. It is a polymer of two saccharides. So we call it a disaccharide, and they are NAM and NAG. NAM is followed by NAG, followed by NAM, followed by NAG, followed by NAM, etc. And if you don't like NAG and NAM, you can call NAG N-acetylglucosamine, and you can call NAM N-acetylmuramic acid. Either one is fine, and they probably mean about the same to you. So peptidoglycan is this repeating disaccharide of NAG and NAM hooked together in a chain. And then we have a chain to the side of it, and then a whole bunch of them on the same plane. And then we have underneath that chain, we have other chains in another plane, and then another plane. And so you can have multiple layers of peptidoglycan. Any question about that? All right, so peptidoglycan does have the disaccharides, but it is hooked together by short polypeptides. Here we go here, that this chain is hooked together by this chain by a short peptidoglycan molecule right there. And then this chain is hooked together to this chain by a short peptidoglycan running right here. And yeah, it's linking up with this short peptide and that short peptide. Any question about any of that? And by the way, this gives peptidoglycan its name. Glycan means sugar, and NAG and NAM are sugars. And it's called peptidoglycan because there are short peptides linking the glycans together. Any question about any of that? Penicillin actually inhibits the cell wall, leading to cell death by interfering with the peptidoglycan when it's being put down in the cell wall. What penicillin G does is it locks this chain being put down, meaning a cell that is actively growing its cell wall puts down new peptidoglycan, and penicillin G can prevent this peptide from forming. And so penicillin G will get in this position here and not allow that peptide to be put in. That actually causes holes in the cell wall. And if you have a hole in the cell wall, the cell membrane can come out of the hole, much like a balloon. If you're blowing it up, can come out of the hole in the box that you're blowing the balloon in. And because there's no cell wall there, water can continue to move into the cell, and the cell membrane will continue to expand out of that hole. And then eventually the cell membrane will burst, and then the cytoplasm will leak out of the cell, and that will kill the cell. Any question about any of that? Now remember, this picture is showing, I'll go to the next one. It is showing the difference between a gram-positive cell and a gram-negative cell right here. Remember, it's only showing 3 layers of peptidoglycan in the gram-positive cell, but in reality, it is hundreds of layers of peptidoglycan, okay? In the gram-positive cell, it's hundreds of layers. I don't know how many layers it is in the gram-negative, but it's more than just one shown here. And it's more than even 3. And this is gram positive, but the gram negative is more than 3 layers of peptidoglycan. Okay, like right here, they're only showing you 1 layer. It's more than just a few. It's less than hundreds, okay, but it's more than just a few. All right, so the gram positive cell wall is mostly made up of peptidoglycan. Here we're seeing it here. A very thick purple layer for the cell wall. The yellow layer is the cell membrane. Peptidoglycan, the main molecule in the cell wall. There are other molecules in the cell wall, such as in gram-positive cells, we have teichoic acid, which run this way. And we have lipoteichoic acid, which also run this way. And the lipoteichoic acid has a lipid portion attached to teichoic acid, and the lipid portion actually binds to and is inside the cell membrane. The teichoic acid portion runs throughout the cell wall. The teichoic acid isn't terribly important, but it may allow ions to help move through the cell wall, meaning the ion can just move right down the teichoic acid. The main purpose of teichoic acid is to help hold the peptidoglycan together. All right, the polysaccharides in the cell wall can provide antigenic variation between cells. An antigen is something that a host recognizes as being foreign to the body, and the polysaccharides in the cell wall can allow the host to recognize the cell as being foreign to the body. Like if this bacteria were to get into your cell, the polysaccharides could be an antigen, which your immune system would then respond to and presumably attack, because it recognizes that this cell does not belong to the body. All right, let's take a look at the gram-negative cell wall. It has a thinner cell wall, a thinner peptidoglycan layer, which is purple right there, and that being the cell membrane, the yellow underneath. And then we have a lipid region above the peptidoglycan that is called the outer membrane of the cell wall. If we look at the outer membrane of the cell wall, we can see that the main molecule is phospholipid, which is the main molecule found in the cell membrane. But in the outermost region of the outer cell wall, meaning the outer membrane of the cell wall, the top layer, we have another lipid, this blue fat lipid, which is called lipid A, and it has a O polysaccharide attached to it. That's that hair-like thing attached to the blue. And so together we call this a lipopolysaccharide. And you can find those in the outer layer of the outer membrane of the cell wall. The lipid A is the important molecule in the gram-negative cell wall, because lipid A is an endotoxin, Endotoxins are something that are toxic to us. And so all gram-negative cells have an outer membrane if they have a cell wall. I'll word it that way. And it has lipid A in that outer membrane of the cell wall. And so all gram-negatives with a cell wall have an endotoxin. Endotoxins are an important topic in microbiology, so we'll be talking about them at 3 different times in the term. And I believe, I might be mistaken, I believe this is the first time we've talked about endotoxins. Is that correct? Have we not mentioned endotoxin before?
That's correct.
So we'll be talking about endotoxin twice more then. All right, in the cell wall of gram-negative cells, we do find more molecules than just the peptidoglycan and the lipid A and the polysaccharide of lipid A. You also find proteins and phospholipid in the cell wall. I think I mentioned this before. The most important protein in the cell wall is the porin protein. And the porin protein acts as a channel to allow molecules to enter the outer membrane of the cell wall, as well as to leave. So if you think about it, molecules like glucose and amino acids cannot get through the lipid bilayer of the outer membrane of this cell wall. And so the cell has this porin protein to allow the molecules that the cell wants, like glucose and fructose and amino acids, to enter the cell wall through the porin protein. Now, for those molecules to enter the cell membrane, they have to have a protein to get through the cell membrane as well. However, in the cell membrane, we don't have the porin protein. What we have is an active transport protein or a facilitated transport protein. And this protein acts to allow one molecule, and generally only one molecule, to enter or leave a cell. So we have a protein, like a transport protein, that allows only fructose to enter the cell membrane. And we have a separate protein, which is a transport protein, to allow glucose to enter the cell membrane. But for all of those molecules, as well as amino acids, they enter the outer membrane of the cell wall of a gram-negative cell through the porin protein. Any question about any of that? All right, so for this slide, you should probably know the porin protein, you should know lipid A, you should know the outer membrane of the cell wall, you should know the peptidoglycan layer of the cell wall, and that's more than one layer. It's only shown as one. I think that's about it for there. Oh, I've already talked about this. I should have brought you to this slide. You do find some other molecules in the outer membrane. We talked about phospholipid is the main molecule. And then you have lipopolysaccharide that would be lipid A attached to a saccharide, the O polysaccharide. And then you also have lipoproteins. These are proteins in the outer membrane, which have lipid portions attached to it. The outer membrane of a gram-negative cell can protect the cell from different things, such as complement protein cannot get through the outer membrane of the cell wall. The complement protein is a protein that we make to help defend ourselves, meaning it attacks cells that the immune system recognizes as foreign. And then antibodies. Antibodies are proteins that we make to attack things, and it's very specific. We're not going to talk about that later. It's a protein that works specifically on one or very few molecules, whereas the complement proteins are a little more general. But in general, both of these attack molecules that the body thinks is foreign. And so the gram-negative outer membrane can prevent those molecules from attaching to the cell, giving the bacteria with protection against complement proteins and antibiotics. And that's applying to certain antibiotics. We'll talk about that a little bit later. But the one antibiotic you need to know that cannot get through the outer membrane of the cell wall is penicillin G, the first antibiotic ever discovered. So if we look at the cell walls, the gram-positive cell wall has a thick peptidoglycan layer. It's a rigid structure. The gram-negative cell walls have a thin peptidoglycan layer, and it is rigid, but it's not as rigid as the gram-positive cells. Gram-positive cells have teichoic acid in them. Gram-negative cells do not have teichoic acid. Gram-positive cell walls do not have an outer membrane of the cell wall, and gram-negative cell walls have an outer membrane of the cell wall. And inside that outer membrane, you can find lipid A, which is an endotoxin. All right. Any question about the gram-positive cells and the gram-negative cells? All right, if there's no questions, let's talk about staining of cells. And we're going to specifically talk about the Gram stain. The Gram stain is probably the most common and useful stain used in microbiology, where it can separate cells into two large groups, the gram-positive cells and the gram-negative cells. How this stain works is that crystal violet complexes with iodine, and then this crystal gets in the peptidoglycan, and in a gram-positive cell, the crystal violet iodine complex cannot leave the thick peptidoglycan layer, and so it will stain the gram-positive cells purple. The gram-negative cells, on the other hand, the alcohol wash or the alcohol decolorizing step dissolves the outer membrane. And that's because the outer membrane is largely lipid, and alcohol can dissolve lipids. The crystal violet iodine complex can wash out of the thinner peptidoglycan layer of the gram-negative cells. And this I'm talking about if you're doing the gram stain correctly. And then we use safranin to stain the cells pink once the crystal violet iodine washes out. Here's a good summary of the difference between gram-positive and gram-negative cells, and you can ignore all the lower portion of the table. Peptidoglycan in gram-positive cells shown here. Oh, we're seeing color too of the gram stain. Purple, pinkish, gram-positive, gram-negative. The gram reaction retains the crystal violet iodine complex, and the gram-negative does not retain the crystal violet iodine complex. A peptidoglycan layer is thick. Peptidoglycan layer is present, but it's thin. Echoic acid is present in gram-positives, absent in gram-negative. The outer membrane is absent in gram-positive, present in gram-negatives. A lipopolysaccharide, there's virtually none in gram-positives, but that molecule is high in gram-negative cells because it's found in the cell wall. All right, any question about the differences? Nope, I didn't switch the page there. Wrong one, that's why. You can make the cell lose its cell wall in different ways. If you lose the cell wall, the cell will be susceptible to osmotic lysis. Penicillin G can actually inhibit peptidoglycan, and so that's why it's in here. You can also get rid of the cell wall by digesting the cells in lysozyme. This is an enzyme that digests the peptidoglycan. And then the cells have different names if it's gram-positive without a cell wall versus gram-negative without a cell wall. But I'm not going to test you on those names, so don't learn them.
Dr. Volpe, I think we skipped a slide.
Say again?
I think we skipped a slide.
Yes, you're right. Let's back up. Let me finish talking about this here. Lysozyme is actually an enzyme that we and other animals make to help defend us against prokaryotic cells, especially bacteria. I was talking about the cell walls. Let me warn you that there are 3 cells that have atypical cell walls. The first I've already talked about is that everything I've been talking about is the cell wall of a bacteria. The archaea, the different domain, have different cell walls from bacteria. Archaea, most of them do have a cell wall, but it is not made up of peptidoglycan. It's made up of pseudomurine, which is similar to peptidoglycan, but it's different. It's another disaccharide. There are some archaea that lack a cell wall, but of course there's some bacteria, just a few, that also lack a cell wall. All right, so that's the first cell that differs, and we're not surprised that these cells differ because they're in a different domain. But in domain bacteria, there are 2 cells which differ, and that's the mycoplasmas. These are bacteria, but they lack a cell wall. Curiously, the mycoplasmas have a sterol in their plasma membrane, which gives the plasma membrane strength and support. And as you remember, I did mention that animal cells have a sterol in their cell membrane to help give the cell strength and then to support the plasma membrane. In animals, that sterol is cholesterol. In mycoplasmas, that cholesterol is not found. It's some other sterol. The third cell, which has a different cell wall, a typical cell wall, is Mycobacterium. It is a gram-positive cell, but it's an unusual gram-positive cell because it has mycolic acid in the cell wall. And it has a lot of mycolic acid in the cell wall. And this mycolic acid is a lipid, and it prevents water-soluble materials from entering the cell or from leaving the cell. The mycolic acid gives the Mycobacterium acid-fast staining properties. So an acid-fast stain gets inside the Mycobacterium and then doesn't wash out. Anyways, Mycobacterium, the archaea, and the mycoplasmas have atypical cell walls. Any question about any of that? All right, the gram-negative bacteria are not as susceptible to gram-positives to penicillin G. Penicillin G gets in the peptidoglycan and then prevents it from being attached with the short peptides. And that is how penicillin G kills gram-positive cells, by preventing the cell wall from forming and putting holes in it. And then the cytoplasm can balloon outside of it, and it will eventually, water will keep moving into the cell, and the cell will eventually burst, and the cell will then die. Although gram-negatives are not susceptible to penicillin G, you should realize that gram-negatives are susceptible to other antibiotics, such as antibiotics that can penetrate the outer membrane and then the cell membrane. An example of that is streptomycin. This is a lipid-soluble antibiotic, and it can get right through the gram-negative bacteria. Another molecule which can get right through the gram-negative outer membrane of the cell wall is actually ampicillin. And if you remember, ampicillin was one of the early members of the penicillin family to be developed. Penicillin G was the first, penicillin V was the second. I think ampicillin was the third or the fourth member of the penicillin family to be made. And it can act, ampicillin can act on both gram-positives as well as gram-negatives. Penicillin G can only act on gram-positives. Any question about that? All right, so let me warn you, when you're talking about gram-negatives, don't tell me that they are resistant to antibiotic treatment, because they're only resistant to penicillin G and other similar antibiotics like penicillin V. They are susceptible to antibiotics such as ampicillin, streptomycin, and other other antibiotics that work on gram negatives, which to the life of me, I cannot remember. Tetracycline would be one. All right, any question about anything? If not, we're going to stop here and I'll see you in the lab at 6:30. All right, bye.